Hello, everyone. This is Chapter 3, the chapter on biochemistry, or the chapter on the chemistry of organic molecules. This chapter is a little bit more of a difficult chapter because it's looking at smaller compounds. It's really kind of tough to kind of make sense and visualize, but we're looking at the four major macromolecules, and there's a lot of basics here of chemistry, and let's just jump into it. All right, organic molecules. This is a molecule that contains carbon and hydrogen and may also have oxygen, nitrogen, phosphorus, or sulfur. But the main elements in an organic molecule are carbon and hydrogens. Oh, by the way, I'm not going to go back to that slide. Organic in the sense of what you see at like a grocery store is not organic in the sense of what we're talking about here. I'm not even going to get to the whole terminology of organic and what that means in grocery stores and blah, blah, blah. Organic in chemistry means, again, it has carbon and hydrogen. Organic in a store means it's a forming procedure. So separate the brain there. Everything in a store, I would say, is organic because it has some sort of carbon in it. Yeah. So organic molecules, carbon has 4 valence electrons like we talked about in last chapter. This means it must acquire 4 electrons to fill that outer shell, because remember that octet rule. This means it's going to create a lot of covalent bonds with other elements. This is where we see a lot of carbons, hydrogens, oxygens, nitrogen, phosphorus, and sulfur. It just bonds with a lot of other elements. So it's really a good element to kind of start forming other structures. And it's the element of life because all life has carbon in it. Let me go back. So carbon skeletons are these straight carbon chains. You can see carbon, 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 carbon. This is our carbon skeleton. So we can make a lot of different structures with carbon. And when you see a bunch of carbons together like this, that's just a carbon skeleton. And what we're going to see is we're going to see a lot of these carbon skeletons in the macromolecules we're going to see later on in this chapter. Notice how, again, let me go back one more time. This is 3.1. So this is the first day of notes. You can see other carbon skeletons here, carbon, carbon, carbons. They can form rings. They can form tons of different structures because they bond with elements, the Chomps elements, so easily. Now, they obviously bond with other elements, but mainly in the macromolecules, we're going to be looking at those 6 elements. All right, functional groups. Man, this is a tough portion because there's just so many functional groups, but the great thing, let me see if I can make my box a little bit smaller. There we go. The great thing about this class is you don't have to memorize these. I would say you should probably know what hydroxyl group is. Leaving this, you should probably already actually know what a hydroxyl group is. It's an OH group, just because we kind of talked about those in the last chapter. And that's pretty much it. During a test, I wouldn't ask you, like, what's an amino group? What's an aldehyde group? What's a ketone group? That's for organic chemistry in college. That's not what we're doing here. But you should become a little bit more accustomed. You know, when you go to college, if somebody says ketone, you shouldn't be like, I've never heard of a ketone before, or I've never heard of a carboxylic acid. Like, you should have heard these terms before. All right, R is used to represent the remainder of a molecule. So you can see we have it in these functional groups, which, again, functional groups, by the way, if I didn't mention, they're just a specific combination of atoms that we see pretty frequently in biology. If you see like the R, so you can see we have an R right here. That just means there's a whole other molecule. It could just be one element, or it could be a whole molecule. It just doesn't matter to us in this sense. It's that COOH that we're looking at for this carboxylic acid. The R is just representing the rest of the molecule. All right, so that's the end of 3.1. Let's look at 3.2 a little bit. Let's go back here. Dehydration synthesis. So think about what dehydration means. You're taking water away. And what does synthesize mean? Synthesize means you're building something up. This is a reaction that binds monomers together to form polymers. Okay. What is a monomer? Think of mono meaning one. It's just a single molecule or a single unit. Polymers, think poly meaning many. So there's many. So this is a reaction that we are going to bond a bunch of monomers together to form a polymer, which is a larger unit. So there's a lot of smaller subunits of the monomers in the polymer, the larger unit. Water molecules formed when H and OH, this is a hydrogen and hydroxyl group, are removed. So you're removing water to form, you're removing an H and an OH to form that water. You're dehydrating it. A covalent bond is formed between these monomers. So let me show you what this looks like. So we have our 2 monomers, monomer, monomer. We have our OH and H. They bond together to form water. We're dehydrating it, and we're synthesizing a larger molecule. This is a dehydration synthesis reaction. Okay. Now, like we talked about with the dehydration synthesis reaction, we also have hydrolysis reactions. Think hydro meaning water and lysis means to break. So we're breaking down this word a little bit. Get it? Breaking the word, talking about hydrolysis. Hydro meaning water, lysis means to break. So we're combining those two, that prefix and suffix, and we're saying, okay, what does that mean? That means that we're going to use water to break down a molecule. So we're going actually in reverse. We're taking a polymer and breaking it down into monomers. In this sense, water is added, and actually the water is split up into the hydrogen and hydroxyl groups to facilitate this reaction. And this is exactly the opposite of those dehydration synthesis reactions, where you're going to add in water and it's going to split these monomers up to form two monomers from the polymer here. This is a hydrolysis reaction. All right, so let me go back here. So we're going to be learning about the four macromolecules next, and these four macromolecules are carbohydrates, lipids, proteins, and nucleic acids. Now, each one of these macromolecules has a monomer, and then like kind of examples of different polymers. So like the monomer for carbohydrates are monosaccharides or sugars. And there are a bunch of different polysaccharides, which we'll get a little bit into later on. But like lipids, there are monomers of glycerol and fatty acids. And an example of a polymer would be fat. Proteins is next. Their monomer is an amino acid, and they have a lot of different proteins or polymers, but we call them polypeptides. And last category are nucleic acids, and their monomer is a nucleotide. And the polymers of nucleic acids are like DNA and RNA. I know there's a lot of information here. Let's start out with saying there's four macromolecules: carbohydrates, lipids, proteins, nucleic acids, and their monomers are monosaccharides, fatty acids mainly, amino acids, and nucleotides. We'll focus on these as we go through each different section or each different macromolecule. Let's start out with carbohydrates first. So carbohydrates, they are an energy source for organisms. You should probably know that because you eat a lot of carbohydrates. They come in a carbon to hydrogen to oxygen ratio of 1 to 2 to 1. What that means is for every carbon, there are 2 hydrogens and 1 oxygen. What I like to remind students, how can I remember this? A carbohydrate, think carbon, hydrate, water, H2O. So for every carbon, there's a water. They're carbohydrates. Now, it's not perfect 1 to 2 to 1 ratio because there's a little bit of chemistry behind how these link up, which we'll talk about. We talked about the dehydration synthesis reactions, but we'll get into that a little bit more in class. Their monomer is called a mono, meaning one, and saccharide, which is a sugar. Polymers are the polysaccharides, which means many sugars. Okay, so let's go over the monosaccharides. Glucose, fructose, and galactose are all isomers of each other. They have a chemical formula of C6H12O6. If you look down here, we have glucose, galactose, and fructose. Now, you might say glucose and galactose look very similar, which they do. Notice how this oxygen and hydrogen, this hydroxyl group, is flip-flopped. You might think, well, that doesn't really change it. It does. The structure of these are really important. One thing we're going to learn about in this chapter and in pretty much every chapter going forward, structure determines function. That's very important. You can see fructose definitely looks different, but what you have to understand is they all have the same chemical formula, but their structural formula is much, much different. It's very different. All right, disaccharides. These are 2 sugars. So they're 2 monosaccharides joined together. You can see glucose and glucose. They go through a dehydration synthesis reaction where we remove water and we get maltose. Notice it's not a perfect 1 to 2 to 1 ratio, but it's very close because, again, you're just adding 2 glucoses together to form this disaccharide. So think disaccharides have 2 monosaccharides. Oh, and there are a bunch of different disaccharides. There's actually more than this, but you can see the different lactose, maltose, and sucrose. They have different monosaccharides, which are determined by which monosaccharides. You can see galactose and glucose, glucose and glucose, glucose and fructose. You can see they have different monosaccharides. And the structure of the monosaccharide determine how they bond together. Again, structure determines function. All right, so one that your book, I'm not sure if your book talks about this one or not. It's called an oligosaccharide. These are saccharides that are between 3 and 10, and I've actually seen some different numbers. On a test, I'll give you an example. I won't say, hey, what's this, what number is oligosaccharides? Is it 5 to 14? Is it 3 to 10? Is it 2 to 8? Like, I want you to know that there's a number above 2, below like 20, that's, hey, there is a assortment of monosaccharides, and they're used for things called glycoproteins and glycolipids. You haven't learned that term probably. We're going to learn a little bit more about it in the next chapter, but understand that this is just to help with cell recognition. You're looking at cells and how they recognize each other. And you can see the different oligosaccharides that are on these cell membranes have different monosaccharides that they are composed of. So they just help your cells recognize cells that are your cells and cells that are not your cells. All right, polysaccharides, many long chains of monosaccharides. This doesn't show you like our amylose, the amylopectin, and the glycogen. They don't show you what the monosaccharides are, and I don't even know what each one of these are like what they're composed of. You just need to understand that they have different structures which regulate what their function is. They can be used for like energy storage. They can be used for structural storage. There's a lot of different uses for these, but they are long chains of your monosaccharides. We got one more slide. Yep. Ooh, let me go back. All right, polysaccharides. These are, again, we're talking about how they can be used. They can be used for structural, like in cellulose. Plants use the sugar molecules to help build build structures within their cells to help them stand up really tall, and that's why they can grow to be hundreds of feet. Animals and fungi use chitin or chitin, whatever you want to pronounce it. Bacteria use something called peptidoglycan, which is also made of monosaccharides. But again, they differ based on their functional groups. Their functional groups determine their structure determines their function. So we are looking at how the smaller parts are regulating their structure, which regulates their function. All right, let's talk about lipids next. Lipids are a kind of a weird group of macromolecules. Their monomer is fatty acids, but only sometimes. I'll get into explain more of that, but this is like the group where like every other group, like it is always amino acids for a protein. This group, it's not always fatty acids, it's sometimes, but yeah, let's just talk about it. They're all hydrophobic. Let's talk about what that means. Hydro meaning water, phobic meaning you can think of scared of, but it pushes away on water. It does not like water. These lipids are just, they do not mix with water. Examples of a lipid are like fats, oils, phospholipids, steroids, and waxes. And I like this meme, lipids when they see water. Yeah, lipids do not like water. Fatty acids, these are long hydrocarbon chains that end in COOH. This is a carboxylic acid or a carboxyl group. This is what we talked about with functional groups. You won't have to identify this, but you should probably know what a COOH group is going forward. Hydrocarbon tails are nonpolar. This means that they have an equal sharing of electrons and they don't have any polar groups. So this is what makes some lipids that have fatty acids nonpolar. And if you remember, polar things like polar things. Polar things do not like nonpolar things. Man, there's a lot of words I'm throwing around here. Polar things, like water, they are attracted to other polar things. Nonpolar molecules are not attracted. They push back on polar molecules. So this is why nonpolar fatty acid chains push away on polar things like water. Now, when we have these fatty acids, they usually come in long chains. You can see the carbon, 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 carbon all the way down here. That's called a saturated fatty acid, where there's just all these hydrogens connected to these carbons. Whenever you see a double bond here, that's an unsaturated. As long as it has one, it's unsaturated. So this is an unsaturated. This is an unsaturated fatty acid. Now you might say, Mr. Fradov, hey, look, there's a double bond right here. Does that mean it's unsaturated? No, that's where the fatty acid begins. That's the carboxylic acid. You have to look at the carbon skeleton for the double bonds. So there's no double bonds here. It's saturated because it's saturated with hydrogens. Here's a double bond. As long as it has one, it's unsaturated. It could have like 10 of these double bonds. As long as it has one, it's unsaturated. What those double bonds do is they actually kind of like bend. No, actually kind of. They do bend the fatty acid chains. And you can see there's a bend right here. And here's that double bond. So we can see straight chain fatty acids that are saturated and unsaturated bent chains here. All right, let's look at a couple of different examples of these these lipids. So triglycerides are an example of a fatty. Let me slow down here. Triglycerides are an example of lipids. The triglycerides, think tri, meaning three, so there's going to be three fatty acids here. The function is long-term energy storage. So what happens is when your body breaks down sugar molecules, so you eat a bunch of like polysaccharides or sugars, and you have too many of these certain molecules, which we'll get into later, they'll reform them into triglycerides, and this is where you store energy. So when you're not eating and you're still using energy, let's say you haven't eaten in a couple hours, but then you go for a run. Where does that energy come from? You're not eating right now. It comes from these triglycerides. That's just one spot, but this is one area where they come from, or one molecule they come from. The glycerol molecule, I don't have you really dive deep into, but understand it's a 3-carbon molecule with 3 OH groups or hydroxyl groups that will form or help bond these fatty acids. So we have a glycerol molecule and 3 fatty acids. All right, phospholipids. These are a type of macromolecule they're in your cells. They make up your plasma membrane. It's a glycerol molecule and 2 fatty acids. The third, where you're thinking, this sounds like a triglyceride. It's very close to a triglyceride. Instead of that third fatty acid, they have a phosphate group. So that's the major difference, is they have a third phosphate group instead of a third fatty acid. Now, the tails are very nonpolar, remember, which means they push away water. Their heads, though, the hydrophilic heads, attract water. So their tails push away water, which means they're hydrophobic. Their heads attract water because this phosphate group has a lot of oxygen, which is very polar. It means it's hydrophilic, which means it attracts water. So these two areas here have different properties. One is very nonpolar and hydrophobic. One is very polar and very hydrophilic. Let me go on. Yeah, let's just go on here. All right, last couple groups here. Waxes. This is it's a alcohol group. It's a alcohol connected by 2 fatty acids. There we go. The alcohol group's down here, and then we have our 2 fatty acids. And these can be straight chain saturated fatty acids, or they can be bent unsaturated fatty acids. All you got to remember here is you have an alcohol group with 2 fatty acids. All right, steroids. This is our last group of lipids, and just a reminder, all lipids are hydrophobic in some way. These are steroid that doesn't have, this is a lipid that doesn't have fatty acids. You can see they're just 4 carbon rings. You have 1, 2, 3, 4. And these 4 carbon rings make it pretty polar, which means it's going to push away on water. So there will be different functional groups that regulate the function of the steroid. So you can kind of think, okay, I've heard that word function before. That means they regulate the structure. So structure determines function. In this case, you have different steroids that can be produced with different functional groups. I think I'm going to show you one here. Yep. So estradiol or estrogen and testosterone, they're both steroids. You can see these are actually hormones. These are very similar in structure, but they're very different too. If you see, we have the 1, 2, 3, 4, 1, 2, 3, 4 rings, but you can see we have a different group here. You can see the double bonds a little bit moved around here, which makes their structure different, which makes their function different. Obviously, having different amounts of these creates differences in people. So obviously we have the structure of the hormone or the steroid determining the function. I'm going to say this a ton in here. Structure determines function. Okay, one more. Yep, we have vitamins. I very briefly go over vitamins. I don't really talk about too much about vitamins in here, but basically this is another type of lipid. It's hydrophobic mostly, yes. I just throw this in here to just say, hey, we talked about it. But there are different organic molecules that are that help with different metabolic processes. All right, let's stop right there for 3. I think this is 3.4. Yeah, 3.4. Right, let's go over 3.5, which is protein. This is probably one of my favorite sections for this chapter. Definitely my favorite section in this chapter. So their monomer are amino acids. They make a large portion of your dry mass of your cells and of your body. This is because you are mostly just a protein. You're a bunch of different proteins, but you are mostly made up of protein. And there are hundreds of thousands of different proteins. I would actually, just for humans, there are millions of proteins out there. There are just proteins. There are just proteins. Everything. It's just proteins are everywhere. What do you call an acid with an attitude? Amino acid. All right. So skipping that meme or skipping that little cartoon, let's talk about a protein. So proteins are made of these things called amino acids. The sequence of amino acids is going to determine the structure of the protein. The structure of the protein is going to determine the function of the protein. I'm going to say this 1,000 times in this class. Structure determines function. So each one of these here is an amino acid. You can see it almost looks like a chain of these little circles. They're like a chain of amino acids. Let's go over what an amino acid is, though. Oh, before we do that, between each amino acid is a peptide bond. This is created during a dehydration synthesis reaction. These peptide bonds are just really covalent bonds. They're just specifically named to be between amino acids and a protein. So if you ever see peptide, think protein. Okay, so now we add color. So we can see that we have different types of amino acids between each or in the protein. You can see there's a sequence to them, and that sequence is going to be determined by a organism's DNA or RNA. Okay, we won't dive deep into that just yet. Let's just talk about an amino acid. This is the common structure of amino acid. You have your central carbon, you have your amino group. These are functional groups if you remember back a couple sections ago. You have a carboxylic acid, which is a COOH, and you have a hydrogen group. So we have a carbon that's central, an amino group, NH2, carboxylic acid, COOH, and a hydrogen group. Now down here, we have that R group. This is the variable side chain. This is just what is different between the 20 different amino acids. There's 20 different of them, 20 different ones. Each one will have the same top portion here. Their R group, or what's below, will be different. Okay. Here is a list of some of them. You can see we have a hydrogen here, which is glycine, CH3, which is alanine. You have CH3, CH3, which is valine. I'm not going to go through all of them. Each one of these amino acids is different because of their structure. Now, how that structure and how this amino acid interacts with other amino acids is going to be determined by what makes up that structure. So you can see we have a sulfur group here, we have a ring structure here, we have another sulfur group and an OH group. Each one of these is going to have different characteristics. And the great thing about this class, you don't have to memorize any of these. If I give you a table, you should be able to tell me a little bit about each. If I say, hey, what's a property of a non-polar group? You should probably say it's hydrophobic. What's a property of a polar group? Well, it's going to bond with, it's going to interact well with water. So things like this. You have to memorize any of these. You do not. I'm going to skip this just for now. All right, so we have 4 different structures within a protein. 4 different almost layers. Our first primary, or it's called the primary structure, this is the linear sequence of amino acids. Now, these linear sequence of amino acids are held together by those covalent peptide bonds. This is just the literal, hey, we have a glycine, we have a valine, we have a leucine. That sequence is known as the primary sequence. The primary sequence determines the secondary structure. What's going to happen is those amino acids are going to start bonding with each other, and they're going to start forming folds. These folds are known as alpha helixes and beta pleated sheets. So those 2 structures, the alpha helixes and the beta pleated sheets, are very specific to the secondary structure. And how they fold is based on the properties of the amino acids, which is the linear to the primary. You can see here we're going from our primary structure to our secondary structure. We have our alpha helixes, we have a beta pleated sheets. There we go. Now, these structures will start folding on each other, and that's going to form our, let me see, go back here. There we go, tertiary structure. So once these beta pleated sheets and alpha helixes start folding and kind of making a shape, that's going to be our tertiary structure. Okay, and how they fold are based on the properties of the amino acids. I'm going to go back a few slides here. This here is a protein, and I can tell this is a protein because we see our alpha helixes and our beta pleated sheets. If you see these little kind of like corkscrews, that's an alpha helix, and these little arrows are beta pleated sheets. And you can see how this protein is kind of folded on each other, and it's kind of just, it's forming some shape, and that we can tell is a protein. Anytime you see this, you know it's a protein. All right, lastly here, let's get to our quaternary structure. This is when we have some proteins that have several polypeptides or several tertiary structures. that bond together. You can see here this quaternary structure below me. You can see there are several polypeptides or several tertiary structures that will form that one protein. Now, not all proteins will look like this, but to give you an example, this is hemoglobin. There are 4 different polypeptides that will bond together to form this protein. This is the hemoglobin molecule. So this is an example of a quaternary structure. We won't go through any videos. Here we go. Last part here that I'm going to go over is something called denaturing. This is when the proteins will unfold because of things like being heated too high or changes in pH. What happens is you break down the bonds between the secondary and tertiary structures, and that will uncoil or unwind or unfold the protein. And when you change a something's structure, you will change its function. And we'll talk more about denaturing in class, but I'm just kind of go over this term just in general for proteins. Last section, which is 3.6 nucleic acids. This section, I kind of really don't go over much of the nucleic acids. I mean, I go over them, but I don't go into really a lot of depth because we come back and talk a lot about nucleic acids in like the later chapters about DNA. So I kind of go through this and kind of give you a kind of like a, hey, this is what we're talking about kind of just explanation, but understand we're going to dive deep into these molecules later on. So nucleotides are the monomers, and there are 3 parts to a nucleotide. You have your pentose sugar. You can see the pentose sugar right here. You have the phosphate group. You can see that phosphate group. I would try to memorize what a phosphate group looks like. You're gonna want to know what those are. And then a nitrogen-containing base. You can see we have our nitrogen-containing base here. So these are the 3 parts of a nucleotide. Examples of nucleotides are like DNA, RNA, which you should know, and actually ATP and ADP. If you remember that term from college biology, you should remember ATP and ADP. That's also technically a nucleotide. So ADP is something called a molecule called adenosine triphosphate. This is where you're going to have 3 phosphate groups attached, not just one. There's going to be 3. And this is like our energy currency of the cell. This is like our energized battery that our body is going to use to transfer energy. ADP is going to be adenosine diphosphate. That's going to be a molecule with only 2 of these phosphate groups. Let me see if we can go back here. Only 2 of these phosphate groups. The energy that is held within that ATP molecule is going to be between the 2nd and 3rd phosphate group. So if we look here, we have our ADP molecule. That's like our dead battery. It has no energy, has no juice, has no electricity. It's just not energized. Our ATP molecule has a lot of energy, has a lot of potential energy, and will be like our charged battery. Here's that example. Here's the showing you that high energy bond between the 2nd and 3rd phosphate group. And I think this is going to end the chapter for today. So yeah.